Foundation Module 3, UH, Modular Integrated Undergraduate Curriculum Physiology, Dr. Sheen, CEO and founder, Sheikh Zayed Medical College Physiology Department. Medical Superfast Simplified Physiology Video Lecture. Notes GUYTON and Hall. 14 edition. Chapter number 5, lecture number 8, page 65. Clarify the role of Goldman equation in generation of resting membrane potential, RMP. The Goldman equation plays an important role in explaining the generation of the resting membrane potential in cells. This equation is necessary for the permeability of the cell membrane to different ions, primarily sodium, potassium, and chloride. The resting membrane potential is the electrical potential difference across the cell membrane when the cell is not transmitting nerve signals, or non-excited state. This potential is primarily determined by the distribution and movement of ions across the cell membrane. The Goldman equation provides a more comprehensive understanding of this potential than the simpler Nernst equation, which considers only one ion at a time. The Goldman equation is expressed as: in a typical cell, the membrane is much more permeable to potassium ions than to sodium or chloride ions. Therefore, the RMP is closer to the equilibrium potential of potassium, but the contributions of sodium and chloride also play significant roles. Role of Goldman equation in generating RMP. Ion permeability or membrane permeability. It includes the relative permeabilities of the membrane to different ions, and different ions contribute unequally to the membrane potential. The cell membrane is more permeable to potassium than to sodium or chloride under resting conditions. This higher permeability to potassium is due to the presence of more K+ channels in the membrane compared to sodium channels. Electrochemical equilibrium. Each ion will move across the membrane according to its electrochemical gradient, which is a combination of its concentration gradient and the electrical gradient across the membrane. Summary. The Goldman equation provides a comprehensive model for understanding the RMP by considering the contributions of multiple ions and their relative permeabilities, it offering a detailed picture of how cells maintain their electrical stability at rest.